All right, welcome everybody to another episode of the Organic Q&A. I do apologize for being a little late today. As you can tell, my color is kind of blown out. The hair isn't as fantastic as it normally is, but we're making up for it with good information. We got our regular host here, Tad Hussey from KIS Organics, and myself, Chad Westport. This is simulcast on the KIS Organics YouTube channel, as well as the Chad Westport YouTube channel. But I want to turn it over to Tad for the introduction of our special guest today. And before we do that, guys and girls watching, I will keep my eye on chat. So if you have some questions, please just go ahead and toss those into the chat. I'll be saving them towards the end, and we'll get the show rolling. So Tad, I'll turn it over to you.
Thanks, Chad. Yeah, so we are here today with my dear friend, Suzanne Wainwright-Evans, also known as the Bug Lady. He is an amazing entomologist, and I've done multiple podcasts with her on the Cannabis, Cultivation, and Science podcast. And one of the things that we were talking about off air was that she wanted to put together sort of an FAQ that people could go to get her most commonly asked questions about cannabis pests. So we thought this would be a great opportunity to just do it live on the show so we can get feedback as well and any other questions if there's time at the end. But yeah, so just real quick, I'm not going to give Suzanne's full resume because it's extensive. I will just say that I firmly believe she is the best, one of the best entomologists in the country, if not the world, especially when it comes to working in cannabis. And specifically, like, she does a lot of stuff with a bunch of crops, but specifically in cannabis, she really is the best. And we're so lucky to have her here. So with that being said, Suzanne, hello, welcome to the show. Thank you for doing this today.
Hey, well, I'm always happy to do anything for you, Tad, to help you out. And Chad, it's nice to meet you. But yeah, it just worked out in my schedule this week to be able to do this. And yeah, just want to help some growers get some questions answered. If they're Harry Potter related, we know they're coming from Mike Wilson. So other than that, we'll talk about bugs, but I can also talk about cats and candy making too, if we need to cover those areas. But
yeah, you definitely have a wide range of interests. That's for sure. Well, let's start off talking about, you sent me a few questions, and I think the first one is one of the ones that I struggle with myself. And I see people all the time struggling with, which is, how do you tell a root aphid from a fungus gnat? The winged versions look very similar. And then how do we treat them?
So I think part of the confusion here was, unfortunately, a bit earlier on in cannabis history, when some people wrote up, they were trying to do the right things by writing up some fact sheets and doing some basic idea of cannabis pests. They got the identification wrong in these pictures. And what they had as a root aphid was a fungus gnat, and what it had as a fungus gnat was a root aphid. And the problem is nothing on the internet ever dies, and these documents never die. And people cut and paste from them. And again, I think people's hearts were in the right place, but I don't believe you have to go to college for everything. I know some amazing people that are amazing in their fields, but I do think with something like entomology, plant pathology, plant nutrition, having the college background really helps to make sure that you are scientifically accurate. And so having these people trying to do the right thing and then the wrong information getting out has kind of created a problem. So again, this is a question I get probably messaged about once a week on Instagram. And the main thing you want to start looking at is, like, if you look at an aphid, they have cornicles off the back, which are basically kind of like tailpipes. It's not an actual tail, which is in the middle, which is the cotta, but tailpipes. Fungus gnats, and this is where I think people get confused. Male fungus gnats actually can have two little structures on the end of their abdomen, and it could slightly be mistaken for cornicles. But the first thing I do is look, is it cornicles or is it the backside of a male? The other thing is to look at the vein patterns in the wings. And this is something that I spend a lot of time doing, especially when it comes to flies. You have to look at vein patterns a lot. And so fungus gnat is a fly. And the fungus gnats are a family, but they all have the exact same vein pattern in their wing, and it actually has a Y-shaped in the wing. And if you see that, where aphid wings tend to be a little simpler on that. So again, I look at see if it's the cornicles are there. I look at the wing venation. Also look at antenna because they're shaped a bit different between the fungus gnats, which have more of these little short ones, where depending on the aphid species, and this is some aphids have extremely long antenna and some have short. Also, aphids tend to be more bulbous, but this is why you need good magnification to be able to look at this close-up anatomy to see these parts, tell these parts apart.
That's great. So is the easiest thing for someone, a beginner like myself in bug ID, is the cornicles like the go-to thing that I should, like if I don't see cornicles, then I can assume it's a fungus gnat? I don't see
You
those
can assume
tailpipes.
it's not an aphid because
Okay.
you can also get sosids, which bark plant lice. They have a body shape like an aphid, and in times gone by, we normally wouldn't see that stuff in greenhouses. But now with a lot of living soils, cover crops and things like that, we can get some of these other insects in there. But the cornicles are pretty distinctive of aphids. Now you do get in there other insects. Cockroaches have like circe on the back, and they can have other little projectiles off the back. But those cornicles are pretty distinctive on the aphids.
Can you give us 2 quick methods for dealing with fungus gnats, as well as like 2 or 3 quick ones for root aphids? Obviously, knowing that fungus gnats are much easier to deal with and much more common than root aphids. And then also like an application frequency to help break that life cycle.
So typically for fungus gnats, the most common method we use is using the beneficial nematode Steinema feltiae. You can also use another predatory mite called Stratiolaelaps gymnotus, formerly known as Hypoaspis miles, but people just sometimes call it strats, and that makes it easier. What these do is they live in the soil and they will kill the larval stage in the soil. They won't do anything for the adults, but from your time of treatment, it takes about 10 days and you will see a decline in your adult population. And how you can monitor that is by using sticky cards. So I like to use sticky cards to monitor my adult numbers to make sure that your soil control options are working. It's very difficult to, there's no spray really you're going to want to do to try to kill adults. You're better off using sticky cards for mass trapping on that. And I have actually up here the sticky card poster back behind me on my wall that I did that actually can help you identify things. If you contact Catch Masters, they give them away for free. But I do have on there winged aphids and fungus gnats on there and how to tell those apart on sticky cards. Now, when it comes to, well, I have to say too that for fungus gnats, some people do drench Natural, which the active ingredient is Bacillus thuringiensis israeliensis. It's actually a toxin of a bacteria that the fly larva consumes, and it basically kills them. It's very specific to flies. It doesn't hurt any other insects or mammals. And that's been an excellent product. I'm super excited because they're actually going to be next year packaging that from mycorrhizal applications in smaller jar sizes, so you don't have to buy massive commercial sizes. Mycorrhizal applications is going to be packaging a lot of these bacterial products in much smaller packaging for use, which will be good. As far as fungus gnats, root aphids go, that's a whole nother thing. There's lots of variables depending on how you grow, how close you are to harvest. But traditionally, what most growers have been doing is doing drenches with microbial insecticides that contain things like Bavaria bassiana, metarhizium. It was Isaria, now it's called cordyceps, but it's basically a fungus that you can drench in. and it will actually grow on the surface of the insect, killing it. And that's been one way. Again, there's, I hate to say 1,000 ways to skin a cat, and there's a bunch of different ways for management, but we have to look at, again, so many variables to decide which is going to be the best option. But the best thing for root aphids is don't bring them in. Have a good SOP for new plant material arriving, because you get them by infecting yourself, generally with new genetics.
Where are they living on the plant? Like, if when I'm doing an inspection of new genetics, I know we should ideally quarantine things. But if I do have to bring it in, what sort of preventative should I do, and where should I look for, larvae and things like that?
Okay, so typically for root aphids, I mean, they get their name root aphid, and we're specifically talking about rice root aphids. Now, I work with a couple different root aphid species, but the one that is in cannabis, and we're also finding it in ornamentals and can be found in some vegetable crops, is the rice root aphid. Obviously, with its name, it likes to be on the roots. And this is what's challenging sometimes is, people want to go to the internet. They want to read about a new pest. When you look at the papers that have been published on this species, the early papers that we all had came out of China. And what the behaviors I'm seeing here does not match what the literature says, because I am seeing this behavior where they will climb up. And we know that when they get to high numbers, or once you're about second week into flower, we see large numbers of them exiting the soil to go find a new host plant. But before that, I'm seeing them climb up into the plant and actually feeding on the foliage and producing offspring, which is something, according to the literature, they shouldn't do, but they are doing it. So you need to, I would dip the root ball and I dip the foliage, monitor it close, to make sure you don't infect them. Now, I'm sure the chat's flying by and I'm not reading it, but I'm sure people are going to ask about about rove beetles and use. Will rove beetles eat fungus gnats? Absolutely. Can they control them? Absolutely. Would I use them in cannabis? No way, not anymore. For years, I've not recommended them because I've had numerous cannabis growers contacting me because their rove beetle populations go so high, they're getting stuck in the buds, and they're contacting me, asking me how to kill the rove beetles, which a rove beetle will eat a root aphid, but it will not control it. Nematodes won't control root aphids. There's really not a thing with legs or worm that's going to control root aphids. Generally, it needs to be some kind of microbial or some other kind of drench product.
That's great. Chad, I'm going to have you take over the questions for a minute. The semi truck just arrived. I have to go unload it real fast. I know. I did see you highlighted a question about from Vlad. I thought that was a good one. If you want to throw that in and then take it away.
I'll do that. We have some good ones here, but I just want to shamelessly plug the soil real quick. Shout out to Rick. Shout out to Chad and KIS. The delivery has been so fast and the nutrient packs are very well placed. So I appreciate your support, Rick. We have a new soil collaboration that has just rolled out through KIS Organics. To the point, Vlad had a question here. He is wondering, and I love this because I need to do something quickly. Once again, they have popped up. But Vlad the Inhaler, he asks, which part of the life cycle is most easily interrupted in fungus gnats? Which maybe my question would dovetail is, what frequency rate should we have for applications?
So you generally go after the larval stage, which is in the soil. Again, killing adults is very difficult. You can mass trap them on sticky rolls or cards. You can catch a lot. But the mites and the nematodes all will go after that larval stage. The mites can also feed on their eggs in the soil. The question of frequency is very interesting. It depends on a few things. And you're going to learn there's a lot of times there's depends.
Yes.
If you've got a huge population of insects in your soil, you apply nematodes, they can reproduce and new nematodes can come out, then you have a little bit of a self-sustaining population. But oftentimes when you have very small hosts, again, fungus gnats, thrips larvae, things like that, they can't reproduce a lot of nematodes in there, unlike a big fat grub. So we do find ourselves reapplying them. There was some really good research done at Cornell by my friend John Sanderson. And what he did is he took pots, put soil in them, and then he applied nematodes to them. And then each week to a new pot, he applied fungus gnat larvae. And up to about, I think it's about 6 weeks out, he was still getting control with the nematodes controlling the fungus gnats. That said, you know, when you release your nematodes, you're going to have a high number, and then the numbers are going to start crashing down. So if you are in the thick of it, it wouldn't hurt to do some back-to-back multiple applications for just the first couple weeks. Then you can back off probably every 3 to 4 weeks and monitor it. Again, use the sticky cards. If you see your numbers start to come back up, do another application. If your nematode numbers are continually going down, I don't see you need to apply them, but make sure, and applying your nematodes correctly is a whole nother thing, because I've seen a lot of people applying their nematodes. They've accidentally killed them. Just make sure you're following all the directions. Don't mix them in hot water. You know, don't let them warm up. Store them around 41 degrees. And every nematode product has different expiration. Like the sponge nematodes, you got about a week. The tray products, you have about 6 weeks to use. So pay attention to that expiration date on there.
And that, I appreciate you mentioning the temperature 41, because commonly that's around what the refrigerator is at. So, you know, anybody needs a reference point there. A lot of people here just on like a home level turn to a product like mosquito dunks, and the active ingredient there is the BTI. You just kind of mix it in with the water. Is there a preference maybe commercially versus going the nematode route versus like a soil drench? Is one more effective than the other, or just a matter of preference?
So the active ingredient in the mosquito dunk is the same thing that is in the natural that I mentioned before. And it's again, the BTI. The problem is it's not a living bacteria you're applying. And so insects can completely build resistance to it. And this is what's happened in a lot of greenhouses, is they used it for years and years and years, and it stopped working. If you're just an occasional user of like, once, every few months, I'm going to do an application, probably nothing to worry about. But if you're commercial production or using them regularly, you absolutely will build resistance to the BT. Nematodes, you're not going to build resistance to. And that's why most of my growers have hopped over and they use nematodes instead of the BT. Also, using nematodes, you also can get thrips larvae in your soil. The BT only kills fly larvae. So you can kill more things with the nematodes where the BT is very specific. Also, I'd mentioned that the natural, my brain, I keep wanting to say dipel. Dipel is the caterpillar one. Natural for the fungus gnats. And I have to do more homework on this, but it was explained to me that the actual volume of active is higher in the commercial product, which again is the natural, over mosquito dunks, which is a homeowner product. So I have to do a little more checking on that. And that's why it's so exciting that with, I think the brand name is going to be Magic Gardener. Actually, I had a magazine article come out just today. I got it, that I wrote a while ago for garden centers about how they should be looking at these commercial bacteria products that commercial growers use, that mycorrhizal applications is packaging now for homeowners in the same concentration that the commercial growers use them at. So that's what's kind of super interesting. And they're doing some different bacillus species for disease and things like that. Because it is frustrating to me. We have some of these amazing products that my commercial growers can use. And when you have a smaller grow, they may not have access to it. But I think the larger companies are realizing that there's a market there. And so they are going through the registration process with the EPA and making these products more available.
That is excellent. And I appreciate you breaking it down that way, because you touched on one of the major principles of, IPM, particularly in commercial, and that's multiple modes of action. You don't just want to stick to one thing, because things will build resistance. And I think sometimes as home growers, that may be something that we could, easily overlook, because, hey, it worked. Why is it not working? So yeah, multiple modes of action, always important to have. So nematodes is going to be very beneficial there. I kind of wanted to swing back to the root aphids real quick. Obviously scouting is your best bet. We're not looking at the roots frequently, but like you said, we often get them when they come in. So at the time of transplant, if I'm taking my little, 4-inch container and moving it up, how closely should I be looking? Like, am I going to see these with the bare eyes? Are they just going to be on the outside of the package, or the soil structure, or are there going to be a little investigation needed to really kind of identify, or to feel good that we're putting them into a fresh pot clean?
So I hate to say this, but it depends on your age, because like when I was in my 20s, I could drive down the highway and see spider mites. And then once I got into my 40s, I'm like, oh, and now I'm in my 50s. It's like, oh my God, I'm, my in fact referred to me last night as like his visually impaired wife, because my vision's pretty bad. But that said, my whole existence has been trained to look for bugs, so I can see them with my progressive lenses, where they've got pretty strong magnification in the bottom for seeing small things. But you should, if you pop them off, you can see them on the outside, but they will be inside the root mass too. You just are seeing the outside one. And this is one of those advantage things. Like if it were a poinsettia grower, we would just take that root ball and pull it apart, because we can afford to do destructive sampling, and we can do that. You can't start doing that with cannabis plants because of the value on them. So it's a lot harder to be able to do that. Also, we struggle with monitoring them, because once they are put in living soil beds, we can't just pop them out of the pot and check the root mass to see if they're there, which is something in containerized plants. We can regularly just pop it off and look at the root health there. So it can be challenging. I would assume that anything you bring in has the potential for having those, or having hemp russet mite. I just assume they would. And I would treat everything like it does, because the cost of treatment is not much compared to having it infest your facility and then having to deal with it.
Very, very true. And so, Tad, I had a follow-up question on the root aphids there, and we didn't really explore too much further, a little bit about the BTI and mosquito dunk. So I just want to let you know, because I know you had some excellent questions that we wanted to get some answers to. So I will let you, if you had another one, toss one in.
Sure. I wanted to hear the answer, because I've gotten asked this one a lot too. With a lot of people growing in living soils, can you have too many springtails in a bed? And I see a lot of misidentification of springtails. So maybe you could touch on that too, Suzanne. People see things moving around and they freak out.
Yeah. So springtails describes a whole group. There's multiple species of springtails. And again, I hate to say this, but I mean, every situation is different, because in certain cropping systems outdoor, they have seen them cause damage to plants, like the globular springtails, even though as cute as they are. And by the way, bucket list this year is photographing more springtails. So that's one of the things I'm going to work on this year. But it depends on the species you have. And again, the numbers of them, generally anything out of check is not good. But what I see happening a lot with cannabis, with springtails, is you have this massive amount, and then they just kind of crash on their own. And I just would be very careful with very young plants, because of the chewing damage they can do on stems. And that's, again, where we typically see the damage is in seedlings. It's not so much on mature plants. And it's a problem. I mean, because they're not really insects and treating them is going to be very difficult. You kind of have to wait it out. But typically we see these springtails and roly-polies and these other soil dwelling arthropods get out of whack often because there's too much organic matter in the soil, and that's what they're just feeding on. Organic matter is great, but it can be too great if you have too much, because then these numbers can get out of whack.
Yeah, I haven't ever seen springtail damage on a plant. That's not, like you said, I can see where it's possible, but I've seen pretty large populations, but it's always been in soil which had mature flowering plants. So I haven't had this issue with young plants.
If you
Have
look
had
up
the issue
for like,
with roly-polies, though. We should probably talk about that a little bit.
yeah,
Isopods.
if you go online and look for, like, again, big agronomic crops and globular springtails damage, there's pictures of it online. And I talked to a researcher who had seen damage out on the field. I mean, it's not a super common thing, but can it happen? But that's also a different one than everybody that sends me the pictures of springtails. It's a different species that people are sending me pictures of. So again, this is where I mean, on my, tombstone, besides it's going to say, don't buy wild harvested ladybugs, the next line is going to be identify everything before you do anything. And you've got to ID things to really know if it's a problem and if you need to treat it and how to properly manage it. I can't believe how often things are not identified properly and people treat. It's a waste of time and money, and people get themselves in trouble. You got to ID things.
Can you talk a little bit about isopods, roly-polies, what we can do?
First of all, you have to have a permit to buy them and move them around in the United States. Because they have the potential to cause damage, as everybody has now learned. And people just are ignoring the regulations because we don't want invasive arthropods here. And actually, a lot of them are now invasive because people moved them. They've got to get outdoors. So first of all, yes, there is a permitting process. Second, when, again, you have large amounts of organic soil, their numbers can go up and they can do damage. I've seen them do damage even in like tomato crops because they will feed on tomatoes. I've seen them chew on the stems of cannabis, and killing them is really difficult. Honestly, I've had growers, I told them to go buy those small shop vacs at Home Depot or Lowe's and just shop vac as many up as you can, because they're very, very difficult to kill. Thing is a lot of soil systems have done fine. It's just, an isopod roly-poly is not a sign of a healthy soil system because they're not native in all soil systems, just like earthworms. There's no earthworms native to the northeastern United States. And people have this idea you have to have worms to have healthy soils. There's lots of other things, other microbes that are doing these jobs. But buying the roly-polies and putting them in, it's just asking for trouble, I think. And I know sometimes people aren't buying them. They just are coming in with soil. Just try not to have such a high organic matter. The cover crops getting cut and drop and putting them on the soil surface, that makes me cringe because of pathogens and recycling mites and aphids, but also that just gives them a perfect habitat to live in.
Yeah, I've always had a few isopods in my beds, and they never got out of control until this last cycle. For some reason, I don't know, because I didn't add any compost, so I didn't add additional organic matter. I think maybe it was some of the protein and some of the organic amendments that I used. Something caused their populations to just absolutely explode. And I always thought like, oh, I don't run mulches, I'm not doing cover crops, I won't have to deal with this problem. But I did. And so what I found worked the best for me, and I'm not saying this is a solution for everyone, was doing mass trapping. So I tried various fruits, potatoes, things like that, even some cannabis leaves. And then I would remove that and as many as I could every day when I went in there. And I was able to get their numbers down quite a bit. I've heard people use sticky cards directly on the soil in empty bare patches, or wrapping the stem of your trunk of your plant with sticky tape as a way to help protect it. But yeah, they do damage the plant. I definitely had like you could see burrows into the stem all the way around, sort of to girdle the stem. But my crop turned out great. The plants did just fine because they were large enough by the time the populations really exploded that the plants were able to, get through it. But with young plants, I think they would have just decimated my crop. And what Suzanne was talking about, just to clarify, I know there's a couple people in the living soil community that say if you buy these certain species of isopods, they don't eat your plants. They're just really good shredders, because isopods in general are shredding up organic matter, somewhat like what worms do. But what she's saying is please don't buy these isopods because we're spreading potentially disease or invasive species around the country.
Or make sure they're licensed and permitted, because the USDA has to inspect to make sure what is being done and sold is okay. I mean, just like biocontrol agents, mites, nematodes, and insects, there's a permitting process that goes through the USDA. They have to check to make sure the species is what you say it is, and it is all monitored to make sure bad things don't happen.
All right, let's move on to everyone's favorite insect pest that I'm sure every grower has dealt with at some point in time. It's all like, it's either spider mites or where I was leading to was thrips. Let's talk thrips.
Yay! I should have worn my Thrips blazer tonight to talk about the thrips. Yes, I have a blazer with thrips all over it. Even though aphids are my favorite insects, I've been doing tons of work on thrips for years now. So, so is there a particular question you have on thrips? Anyone?
Well, so thrips damage is fairly distinctive. I would love to hear how you describe it on a leaf. What are the identifying characteristics? I feel like it's kind of like two-spotted, like it's one of the easier insects to identify, but I also think it's kind of one of the harder ones they're persistent. Like they're hard to like completely feel like you've gotten rid of them. Like you can control them, but they stick around. I don't know.
Well, yes. Why are the thrips assholes? That's the best comment of the night. I like that. So they're just
Let's
they're
start with
just
the
trying to make
damage
a living.
and what they look
They're
like.
just
Let's start there.
okay. So thrips are typically cigar shaped. They are one of the few insects that have asymmetrical mouth parts, which I have a brace on my finger, so I can't do my normal mouth party things. My finger will bend. But they are different. So spider mites puncture a cell and feed on it. Aphids have a very long mouth part like a straw. And so they tap down into the plant and like hit the veins in there to feed. The thrips kind of do this rasping on plant cells where they kind of rasp and then eat the contents out. And when they do that, they're removing the chlorophyll. And that's what can cause kind of that silvering that's left behind. That said, different thrip species cause different kinds of damage, which, if I were to talk about, my pet peeve lists of the cannabis industry is identify your thrips 2 species before you have a program, because to this day, I still see people saying they have Western flower thrips. I'm not going to say Western flower thrips is not in cannabis. I know it is on very rare occasion, but I have yet to ever see an actual Western flower thrips in cannabis. And, Tad, when you said, oh, identifying thrips is easy. Well, knowing it's a thrips, getting it to species is not. I don't even think I have it. Oh, I may have it here.
Knowing that you've visited hundreds of facilities and you've seen thrips in so many places and you've never seen Western flower yourself, what species are you seeing? Is there more than one? And is identification beyond like too difficult for the average person like myself, or am I just going to be saying thrips and that's going to have to be good enough?
So most of what I see is onion thrips. That's what I've been seeing. But also I've seen Echinothrips americanus, which is a Native American species. I've found out on the West Coast, there's a genus Caliothrips, which now I'm finding on the East Coast, and it's only in greenhouses, and I think it moved on plant material. There have been reports of chili thrips from a a person that I trust to have reported that correctly, but I've not seen that. So I think that's a more rare one. But now we have a thrips that we have found in the Midwest, and it hasn't even been reported since like the 1960s. And its host plant was hops, which is kind of, usually we don't have things hopping between hops and cannabis, even though they're closely related. So ID is kind of important because a kind of thrips, which can be rare in cannabis, but is very common in other crops, you treat that differently than you do onion thrips. Can you identify that? Like, I think what's happened is people look at the picture, and since overall worldwide, the biggest thrips of economic importance is Western flower thrips, people just look at the pictures and try to match. But literally, I take these thrips, I put them on slides. One of my microscopes is right back behind me here. And I literally look at the hairs on the pronotum, which is the base of the neck. I count antennal segments. I have to look at all these parts of the anatomy to know what species it is. After looking at thrips like this for decades, I'm better at identifying them. But, in the field, if you just handed me a thrips and said, is it onion or Western flower, I'm not going to be able to see well enough because of the hair pattern differences in there. It's kind of semantics because, it's not, it is and it isn't. Because I feel it's very important to identify it so we can do the right program. That said, when it comes to Western flower thrips and onion thrips, the programs can be very similar for management. So sometimes people kind of luck out without doing the identification on there. But if somebody walks in and tells you you have Western flower thrips in your cannabis facility and they haven't put it under a scope, I would be very skeptical of doing that. Yeah, so I'm looking at the thing you just put up, like the ocelli, which are between the eyes. They're like false eye spots for detecting light. That's one of the things on onion thrips. They're more gray. On Western flower thrips, they're red. You just hope that you have your thrips laying right. And you can see from these pictures, these are obviously taken through a microscope. Again, I also look at the hairs and the promotums and again, count antennal segments, but it looks like the onion thrips on the left, its antenna either got broken off or they're not in there. So working with somebody.
Oh, so the antenna is not a differentiation, because that's the only thing that I can tell between these two besides the coloration is that one has antenna and the other doesn't.
They're broken off. That's not a good sample. They're broken off on there.
Okay, so we're not going to be able to identify the difference between onion and Western flower, or maybe even
Probably
echinothrips.
not.
What can we do as a grower? How would we should just, if we were just going to assume that we have onion thrips, just because we may not have access to an entomologist, how do we proceed?
If you look at it and you think it looks like the matching picture of Western flower thrips online, it's probably onion thrips. Echinothrips look completely different. The caliothrips are striped. They look very different. So just assume it's onion. And again, during COVID, I had people sending me tons of samples. Thank you, everybody that did. Onion, 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 all day long. Okay, so onion thrips. First of all, unless you have a crazy amount and you don't do anything, it's just going to make your leaves look like shit crap. Trying not to swear. On the foliage, but generally the onion thrip's not going to be enough of economic damage. People will spend insane amount of money to try to not have them, and it's really eating into the bottom line. And I don't know anybody who has just extra money to throw away. Would I take steps to minimize them? Absolutely. I think for cannabis, for the average growing situation, again, it's hard with temperatures, climate, how people grow and this and that. But I think having cucumeras is the backbone of a program. It's the predatory mite. It will feed on onion thrips and Western flower thrips. It also feeds on broad mite, another problem in cannabis, and it will snack on two-spot spider mites. So it's doing multiple things for you. It's inexpensive to use. It's produced right here in the United States at Beneficial Insectary. You have other European suppliers of it as well. And that could be a good backbone of a program. why I see people ending up with thrips problem is they wait too long to treat. That over and over again, when I talk to people, what are you doing in propagation? Nothing. And even sometimes in veg, nothing. And they're waiting till they have a problem. In ornamental and vegetable crops, the second there is green, either it be from seed or a cut, we are putting out bios on it from the get-go to stop the problem. And so you can use cucumaris on the foliage. It will help snack on thrips eggs a bit. There's some research showing that now, but it will really go to town on those first instar first instar onion thrips once they hatch out of the leaves, and they can kind of snack on the next life cycle stage, but they cannot control the pupa or the adults. But since you're using nematodes in the soil, the nematodes in the soil will get that pupa. And if you keep plugging away at those early instars with the mites, you can really help break that life cycle. And that makes for a very economical program out there. Aureus is very famous for feeding on them. The challenge in cannabis is, in order for Aureus to really be successful and establish, you have to have pollen in their diet. And so if you're a pepper grower, they work great. You put them out, they feed on the pepper pollen, they feed on the Aureus, they feed on mites, they do all kinds of things. In cannabis, you don't have pollen for them to feed on. And also you need 12 hours of light to keep them from diaposing, depending on how you're growing, where you're at. So if you have less than 12 hours of light on a plant, the next generation of Aureus will go into diapause, which is basically hibernation. And so they won't really do well. And it gets expensive to try to dump out Aureus every single week. I think you're much better off because you just need to suppress those thrips using cucumaris and nematodes. If you wanted to come at it from a spray standpoint, every state's different. But the microbial products, which we talked about before, Bavaria bassiana is one, which I'm super excited. Bioworks product principle just got registered on Friday, which is the new Bavaria bassiana product. And I'm super excited about that. But you can use that. Soaps can work very well. Again, what crop stage are you in? What state are you in? There's lots of variables. Soaps can work well to knock back those early instars and adults. EpiShield, which actually came to the market more as a miticide, we've actually been getting really good knockback on thrips with it without phytotoxicity, as long as you use it as the right rate. It's such a low use rate that it's easy to overmix it and you could burn your plants. But the EpiShield's been very good for a knockback. And at the same time, you can take out your spider mites with that too. I still always love Suffoil X. It's a great product to knock back adults and those early life stage. So when somebody calls me and says, I've got thrips, are you a commercial grow? Are you a home grow? What's your media? How far are you from harvest? What are your temperature? I have to look at all of these things to decide what is the right answer for somebody, because there's not one silver bullet answer for everybody. So it's kind of tough, but the products and the bugs I talked about, using combinations of those, will help you with your program. And all those things we know work for thrips. There is spinosad. And this is another interesting thing about the identification. Spinosad, which some people will use, some people don't use. The active ingredient is approved for use in organics. Generally for Western flower thrips, I don't want to say there's total resistance, but there's a lot of resistance. So we generally don't use spinosad for Western flower thrips, but it still works on onion thrips. So if you know it's onion thrips, you can get a better kill using spinosad. But if you had Western flower, it's probably not going to work for you.
So I want to share what I know about thrips, and you can tell me where I'm wrong, because that might be easier as a grower. Okay, so you did mention this. So they lay I guess we'll just talk about onion thrips, since that's what we primarily are seeing cannabis. They lay their eggs inside the leaf, right? So if I get a clone from someone, there could be eggs in there that then become an instar, which is still on the leaf, and then it drops to the soil and becomes larva, or does it all stay on the leaf?
Okay. This is why PowerPoint is good, because I could show you this stuff. So we'll pretend here's the leaf. And here comes the adult thrips in. Insert the egg into the tissue. Up until this last year, from a spray standpoint, we thought we couldn't kill the eggs, but work by Michael Brown Bridge found that a Suffoil X actually can kill about 50% of, they did this on Western flower thrips, but again, very similar to onion thrips eggs, because even though you can't see it, those eggs are in there and there's a little breathing hole because the eggs still have to respirate. And if you plug that hole with an oil, it suffocates them. We also know that predatory mites can stick their mouth parts in and kind of get them right there too. That's how we can kill some of the eggs. But nobody kills it. Let's say, okay, then here comes the larva out. And right now you have a newborn, no wings, no nothing, and it's just going to climb around. That is what we refer to as the L1 or newborn. It's going to molt after it's fed, and you get a little bigger one with no wings, and it's called the L2 or larval stage 2. This is basically a toddler. And so this is still going to walk around. Again, it's going to rasp on those leaves and feeds. No wings. Those are that really light, creamy stage. Next, it's basically going to have to go through puberty. So you get what's where it's going to get ready to molt. And so then it will go into this like pre-pupal stage, and then it's going to drop to the soil. And in cannabis, a lot of them, if you've de-leafed a lot, it can make to the soil. When you do work with like cut roses and things like that, actually a lot can get cut in flowers or in the crotches of plants. So this is where I look at plant structure too, to see where they're pupating. So they're going to go to the soil. They're basically 17 when they hit that soil. They're going to go through puberty and all their changes. And when they pop out of the soil, they're going to be 18 years old. They're going to have wings. They're going to be able to sexually reproduce. And then they fly back up onto the plant and start feeding, mate, lay eggs, and do that whole cycle again.
Okay, got it. I guess I don't see the flying stages very often, or at least I haven't in my grows. I just see the sheen damage.
Those are where you actually see the wings and have the darker on the back. That's commonly what people see. And if you do, and I need to get a video done. I'm teaching this at all my hands-on workshops, but how to wash cuttings, you'll find all the different life stages. And it's a much better way to scout because flipping leaves is not a very effective way for scouting for pests. But plant washing, which, plug for our workshop, Tad, out in Montana. I'm going to be bringing all the supplies and teaching that there for that. But I probably should get a video done on that. Teaching growers how to do that because it's a way to survey for them better.
So yes, September 5th through the 7th, Bozeman, Montana, Cannabis Cultivation and Science Conference. Chad, you're invited. Obviously, it would be great to have you there too.
Yes.
Suzanne's going to be doing a workshop, and she's going to be giving a talk. There's going to be a bunch of people from the podcast there. It's at a really amazing facility. Information at kisorganics.com. Okay, that was the shameless plug. Getting back to the thrips. So what I did when I got cuttings, and the guy who gave them to me goes, "Oh shoot, I have thrips. I just gave you thrips." I did a Suffoil X dunk or dip. What you call a dip at the Suffoil X dip rates based on the Canadian label, and fully dipped my plants. I released those beneficials that you mentioned along with the nematodes, and I saw a little bit of damage throughout that grow. And then I started my next cycle. I did the same process, and I didn't see any thrips damage at all that cycle. I won't say I eradicated it because I know you hate that word when it comes to pest pressure, but I was unable to find any thrips pressure that second cycle. So that was a very effective way for me to deal with it. I love the idea of using some sort of knockback like a Suffoil X or EpiShield. And you did mention that Suffoil has that ability to coat the leaf in a way that may be more effective than some of these other methods, specifically for thrips, which I think is really interesting. Was that a good way of handling it, or do you have anything you want to add, or should we move on to another insect?
Yeah, well, I mean, I think using the sticky cards to monitor for their numbers for the adults, because the sticky cards only work for the adults, it's a good way to know if they're around, and that's typically, how we monitor their numbers. I mean, I am forever, bringing sticky cards home with me from growers, counting, looking to see what's going on. And then that gives me a snapshot in time of what's happening on the plant. I understand if not wanting to do sticky cards because it's one more thing to do. Cannabis growers way overuse them. You just need a couple out to monitor. It's good to keep them by your doors, and then change them weekly. And it will help you monitor for them. Again, for massive thrips issues, people will put out those large yellow sticky rolls, which we can do mass trapping with too. But yeah, I mean, again, the problem is there's so many ways you can manage thrips, and you kind of have to look, because if you are, let's say you're in a living soil bed or even in pots, those thrips can drop to the soil. But if you're growing in the bricks of cocoa with the plastic wrapped around them, like in a hydroponic system or in rock wool, which people do, those thrips are going to fall and hit the plastic. So nematodes and Stratolaelaps aren't going to help you with those, and they can still pupate just laying on the plastic. So this is why you have to look at every growing situation and try to hobble together which is best for what. And again, I wish I could give one easy answer for everything, but unfortunately there's not.
Chad,
I do always like. Yeah, thank you. I always like, it depends. That's something I preach on this channel. So I'm glad that people are hearing it from someone other than myself and Tad. Real quick though, to just kind of the novice eye, would somebody maybe confuse the flying thrip for a fungus gnat? Just be like, oh, I've got fungus gnats. No, okay.
The size difference is very different, and it's very rare that people can actually see thrips fly. I will say I was doing a photography thing to take pictures of sticky cards and cannabis, and this one company's cards, I was putting them out, and then I was actually watching thrips fly in on stick on them, and that's very rare to see, but they were super attracted to that yellow. And well, it was Catchmaster cards. And so, I mean, they did a phenomenal job, and that's when I kind of got sold on those cards. And
yeah, I want to touch on that really quick, because you introduced me to Catchmaster. I learned some stuff that there's differences in the adhesive that you can use where, because I remember back in the day, and this is probably like 10, 20 years ago, you would get some card and it would get stuck to your hair or get stuck to your hand, and that adhesive was like nearly impossible to get off. They've come a long way. Like the Catchmaster cards that I have now, if I get them on me, I could just wash them off with a little bit of soap and the stickiness is gone, which is really nice. I think they make really high quality products. But I wanted to talk to you about, like, where do you put your sticky cards in a small grow? Let's say a tent grow, like what Chad has behind him. Where would I put my card?
So, what I would do, you kind of think about what pest you want to target, but here, I'll just use my sticky card. So if this is the soil and you're very interested in a fungus gnats, I would stand it like this on close to the soil surface. Some of the cards do come with little picks you can stand them on. A lot of my growers will use dowel rods and put a clothespin on it to hold it. For thrips, you actually, if this is the top of your plant, you typically want them right above your canopy up here, because since the thrips are typically flying back up and flying around, then you can catch more of them up top on that. So
would you see, I've always put mine inside my canopy. Is that not the best place?
Pain in the ass to do that. If you want to make more work for yourself, get leaves stuck on them, you can. But you think about where they're coming and going from, and I think on the soil for fungus gnats and even some of the thrips coming out, but also just above the canopy of the plant for flying thrips, because also you're trying to get, if thrips are coming in through your vents, for those that grow in a greenhouse system and not an enclosed grow, you're trying to catch ones coming in, and if you have them up above the crop, new ones coming in will go right there.
That's great. Oh, that's a good pointer.
There was one last questions on the thrips.
Sure.
And I know you guys could probably speak to this. How can ladybugs help rid thrips? Is that a viable option? Is my addendum to this question. Is it a viable option?
No.
Perfect.
Stop with the ladybugs. Stop. I mean, Tad has a wonderful podcast on it. I've done some interviews. The wild harvesting ladybugs is terrible for the environment. They really are not efficacious. What is interesting and exciting, though, up in Canada, Grow Live is now producing Comia maculata. It is one of our North American native species, commercially produced inside of a building. So they don't take anything from the outside. I've been to the production. It's amazing. So we are starting to get some commercially reared ladybugs that don't take from the environment and that are a bit more specialist. Also, there's a newer insectary coming from Italy. I mean, newer is, and they've been in business about 25 years, but coming to the U.S. market. And they have some other ladybird beetle species I'm very interested in looking at. So the commercial rearing portion of biocontrol is responding and looking for some of these other ladybirds that can help with some of these other pests. But typically, ladybirds are not big thrips feeders. They like aphids. Some of them feed on spider mites. Some of them feed on whiteflies. And also, they're heavy pollen feeders, some of them.
And native populations and attracting that is wonderful. Probably not for your indoor, for cultivation, but for outdoors, that's fine. Don't go harvesting your outdoor ladybugs. You'll probably kill them. And it's not a good option anyway. Can you pop up this question here? Because I thought that was a good one worth answering. Michael wrote, I thought sticky traps were just for letting you know you had bugs. Not really good for control is what I heard.
Okay.
Can you touch on that, Suzanne?
Yeah. The cards are typically used for monitoring, but the rolls or the bigger cards are used for what we call mass trapping. And so this is where, you look at the size of your facility, because again, when I'm in like, a 50-acre greenhouse, a little sticky card, every 6,000 feet, it's not going to do much for mass trapping, but putting the rolls way above, we can trap a lot. If you're in a tent situation, though, throwing in a bunch of cards, you're probably going to trap a fair amount of fungus, gnats, and western flower thrips in there. So you have to think about the size of your space. And even though the sticky cards are designed, again, for monitoring numbers, every one you catch is one less one having babies. So, yeah.
Sorry, I was laughing because of the Ladybugs movie joke. I thought that was pretty witty.
No, I'm not even looking at all these things.
No, it's good. We're almost out of time. There's so many more questions I have for you, but I do want to touch on this last one. Where to buy beneficials? Because I think people struggle with that, and I think some of the most easily identifiable. There we go. I got stuck there for a sec. On Google are not necessarily the best. Personally, I mean, and this comes from someone who sells beneficials, so take this with a grain of salt. But I wouldn't want to support any company that's also selling wild harvested ladybugs, because there are a couple that do that too, and they know better. So shame on them. Where do you recommend people go to buy? Because first of all, there's not that many insectaries. I don't know if a lot of people know that. Like Arbico's not rearing bugs. Where should we be looking to get our beneficials?
Well, I do see Q-Trax ask. He says for UK, please. Just so if you in UK, contact BioLine, because BioLine has production in the UK. I just happened to see that. I am not playing inside. I spray. You're going to spray yourself into resistance problems, and that's why everybody's using biocontrol these days. Just as a comment, but back to how to buy beneficials. So, yeah, BioLine is an insect producer in the United Kingdom. That's where they produce bugs. Got it. Okay. They're, I think, in Little Hampton. I know they're just east of London because I've been out there a few times. Anyhow, so in the United States. So typically, if you just Google it, a lot of those companies are distributors that buy the bugs, bring them to their facility, slap their label on them, and send them back out. They're very good at search engine optimization. They're very good at website building. I love all my bug people that produce bugs, but they're not always the best marketing and business people. And so oddly enough, a lot of times when you look this stuff up, the large insect producers in the world, which the biggest producers are Copert, BioBee, BioLine, BioBest, now BioPlanet, and Beneficial Insectary. Those for the North American market, I would say, are who are producing the bugs. The challenge is finding somebody that can get you the bugs fast enough without a lot of stop-offs on the way. And I think I always feel like I need t-shirts, and I need a t-shirt that says, biocontrol is like sushi. You don't want it a day old. You want to have your beneficials as fresh as possible. And this is where, when people call me and they say, hey, I want to buy bugs, who is going to be the best supplier for me? I do not have one universal answer, because what I look at, Tad, always buy from
okay.
Tad.
Well, we're not always the best, but we do ship direct from Insectary, overnight shipping.
Yes.
Which is what you're getting around to.
Yes. So this is the things I look at. I first look at is like, where are they located? And what beneficials do you need? Because not all insectaries produce everything for everybody, or do they have all the same packaging? And then I look at, okay, who can get it there the quickest? So I have no issue when you're working with a distributor that drop ships direct. So if you place your order with Tad, it's not like the bugs are going to go all the way out to Washington and then come all the way back to the East Coast. He works with an insectary, with BioBee, and you place your order with Tad, and then they drop ship right to you, so you don't have them being touched. A lot of, the internet distributors, the product goes from the insectary to their facility, and this is like, how long is it there for? And they will tell you they have perfect climate and and that's, can be okay, but statistically getting it to you fastest and most direct is going to give you the best quality product. Certain beneficials travel better than others, like cucumaris. It's got some food in there. If it's kept at a moderate temperature, it'll be okay. But things like Priscillus, they don't travel well for long periods of time. So if you get them, you need to get them out. So again, these are all these variables I look at. I do think that when it comes to servicing the cannabis industry, I will just say, I think some companies have done better than others with doing that.
One other thing you taught me too is that there's a minimum viable amount of insects, sort of like a critical mass to ship. Like that's why you can't just buy like 100 SF nematodes, because they won't survive the trip with very good viability.
Right. Like with nematodes, if the packaging gets smaller, it's harder to keep the optimum humidity and temperature. And so the actual quality goes down. So that's why there's certain minimum packaging size, because it can have to do with the volume needed to maintain that during shipment. But at the same time, when I was talking with one of the research entomologists at one of the insectaries, they had really good success shipping the minute pirate bug. And so they wanted to ship more. So they just doubled the number in the bottle, and then they all arrived dead. And that was because there was too much CO2 emission in there from the insects respirating. So all the different insectaries have the research that has gone into shipping beneficials is insane. And there is specific reasons why they do the things they do. You have to have enough oxygen exchange. You have to maintain the right humidity without getting condensation in the packaging. And so that's why a lot of things are packaged the way they are. So.
Yeah, if you're new to beneficial insects, I would say reach out to someone that can help you get the right ones for your grow, whether you have a tent or you have a facility, because it's really easy to get the wrong ones or the wrong quantities where you're either overspending or under applying, and so yeah, it's complicated. That's the one thing that you've taught me, is that it's very complicated. And so yeah, just I think that's the one pointer that I want to add to the conversation. I don't know.
I know that the answer is it's going to depend on what exactly we're working with. But what is there a general best way to distribute your beneficial insects? Is it leaving it hanging in the packet on the branch? Is it sprinkling over the plant? Or is that just too broad of a question?
That's way too broad of a question. But I will. So nematodes, you can use a watering can, backpack sprayer, get them on the soil, try to wash them in. Works great if you add a surfactant with the nematodes in the water to get them down, settled in. Nematodes. When it comes to predatory mites, some come in sachets, not all, that you can just hang on the plant. What's nice about that is it gives you a nice slow release of mites over 4 to 6 weeks. And so, but you have to get those in early before you have a problem. Those are mainly used for targeting things like Western flower thrips and broad mites with things like Cucumaris and Swirsky in there. Persimolis doesn't truly have a true slow release sachet. That comes in a bottle, but my favorite way to apply those for cannabis is in the vial with the nipple top. It looks like a miniature ketchup bottle. And Persimolis have this behavior about running up. And so when you hold the vial, they run up to the tip and you can just touch it onto the plant. Sorry, I'm not giving you the birds. My finger doesn't bend, at least this hand. So you walk along and you touch it on the plants and they go running onto the plant. That way you get no carrier on the foliage. Because if you try to sprinkle a lot on the plants, you lose a lot to the floor. You can get vermiculite stuck in the plant. And we kind of, want to try to avoid doing that. Now, granted, my growers have to treat like 50 acres. They're going out with blowers, drones, all kinds of stuff. And they're just putting out so much volume of it that even if some fall to the floor, it doesn't matter. But when it comes to cannabis, that vial with the nipple top for Persimolis is my hands down way to apply them. When it comes to lacewing, you can buy lacewing eggs glued on little cards, and you can just walk by and hang one on each plant. I use those on my house plants, but if I were a cannabis grower, I would be on that lacewing program because the lacewings will not only feed on aphids, they will also feed on spider mite eggs, and they're a good generalist predator, and they're very inexpensive to use. That would be, between that and Cucumaris, that would be like the backbone of any bio program for cannabis out there. So those are kind of the main ways that you would apply the most common beneficials. I saw somebody on there about Persimolis in the heat. So what's interesting about Persimolis is I've worked with Persimolis since the mid-90s. And in Florida, where we can be like 105 in a greenhouse, we're still using Persimolis because the humidity is high enough. There's this weird thing that when you look at the temperatures, they often don't take into consideration humidity. And Persimolis, I think almost humidity is more important than the temperature, because if your humidity goes low, your eggs don't hatch and they're just not happy. But if you have enough humidity right on that leaf surface, the Persimolis can handle those higher temperatures. And even if it's super hot in the day, they go down in the plant canopy and then come back up at night at work. Because essentially the predatory mites are basically blind. And so they're not on a 9 to 5 shift like we think that. So a lot of times they're running around at night and they'll be feeding when the temperatures are cooler. You can't always go by those temperature charts that are given out from the insectaries. I've asked sometimes, where do they get these temperatures? And it's just, I think there's been a lot of cutting and pasting on temperatures, because even I was an advisor on this research project at Purdue and looking at beneficials for cool season aphid control in high tunnels. And they found that the lacewing larvae, they can freeze them for 30 minutes and then wake them, take them out of the freezer, and they go on their way and start feeding again. But if you look at the temperature charts, according to the literature, they don't do that they would die. But no, they work in them. So I think sometimes it's really, you almost have to test them in your situation with your day and night temperatures. You have to look at your humidity. You got to look at leaf surface, because that's the thing. Once you get that trichome developing cannabis, a lot of the beneficials just get gummed up in it. And I firmly believe coming in with a cleanup, with your dipping of your cuttings, starting right on bios, staying heavily on bios up until flower, and then let it ride, should be how you do it. Because if you spend a ton of money dumping out beneficials in flower that just most of them get caught in the trichomes, and it's a waste of money.
I'm glad you touched on that, because that's definitely a key point. Is all of the biocontrol options are best used in veg early on in the plant life cycle, because they're going to get into the places that your sprays necessarily can't reach. They're going to be working 24/7. The bugs can't develop resistance to them, as you've mentioned, because it's not like a chemical application. So they're a wonderful tool. Well, we could keep going for a while, but we've already gone past our time. Thank you for coming on today. Maybe we can do this again sometime, because I know there's a lot more we can always talk about. Or maybe some people will come out to Montana and come hang out with us in person.
Yeah, I was like, the join the chat thing. I can't connect my YouTube thing because I have like 6 YouTube accounts somehow, and it's all messed up with Google, so I can't even. But I'll get that worked out by next time.
Okay. Yeah.
Get the
sorry, I'm just like, I can't like looking at these comments and like, I want to answer all these questions and comment on them. You know, Captain Jack's dead bug, that is spinosad. So we did talk about that on there. That's just the homeowner version of conserve. That's the homeowner label. And somebody said that they just got alcohol and wiped down every leaf on their plant. How does anybody have that much time in their day? That's just
you can get lost
home.
in the chats. Here's the conference, one of our things there, but
yeah. Oh, as I click off of it, let me click onto it. But I wanted to.
Yeah, and for the conference, I'm going to have beneficial insects there. We're gonna go through kind of how to QC them. I'm gonna go over how to apply them, and we're gonna do like hands-on visual stuff. So it's going to be a good, and this way you can see it instead of just talking about it.
Yeah, there's all the information there. It's going to be a lot of fun. Third day, by the way, of the event is going to be a tasting event. Montana Living Soil Alliance is donating a bunch of weed for that, and we're going to have a cool, fun hangout. So people, if they want to experience what the cannabis industry has to offer in Montana and what Living Soil has to offer, it's a great opportunity for that too. So it's a 3-day event. It's going to be a blast, and we look forward to seeing people there, hopefully. And thank you for your time today.
Yeah, thank you so much for having me. I'll definitely, this was super easy peasy. I did take a shower for it. That's all I had to do work-wise today to do that. But no, I'll be glad to come back and do this again when I have some free time, and we can talk more in depth, because again, unfortunately, it's complicated, especially if you don't know the terminology. And I don't want people to get lost in the weeds, but as Tad said, it's best to get somebody that can work with you, that can just handhold you through it, and then they send you on your way and you're good.
Yeah, there are some videos on our YouTube channel, on the KIS Organics one, on hemp russet and a couple other insects that we did, I think, during COVID together, as well as a microscope video. You guys can check out some of that content if you want to see more stuff from Suzanne. She also has a YouTube channel. If you want to plug that, I haven't looked at it recently. If there's
I haven't put you know, I'm trying to get stuff done, but I generally am on a plane every week going somewhere. And then when I think I have some free time, there's always a crisis somewhere that I am.
Okay, bugladyconsulting.com is her website. You can go there to check stuff out. And,
or Instagram.
Or on Instagram.
I do have both the website and the Instagram for Suzanne down in the show notes, guys. So very easy. Just scroll down, click, boom, right there.
And yeah, we'll do this again next time. Maybe we can talk a little bit about hemp russet and field some questions from the chat. But yeah, this was fun.
Absolutely.
Thank you.
Yeah, thank you guys both for making the time today. Sorry to interrupt you there. I just wanted to thank everybody in chat for the questions, for hanging out. Please, if you have more questions, put them in the comments of this episode and we can get to them on the next time. But greatly appreciate your time today. Thank you so much for coming. And any last words, or I'll hand the floor over to you if you would like.
Oh, me or Suzanne?
I don't know.
That was
good.
sorry. That was to our gracious guest. But Tad, you can have the floor too.
I'm good. I got a lot to do today, so I'm going to get back to it. But I had a lot of fun. It was great to see you both. Thank you.
Cool.
All right.
Have a great
All
day,
right.
everybody.
Thanks. Talk to
Bye-bye.
you later. Bye.